Right? And April is so scary. Like, how can we get possible? Like, I know what you
Okay, let's start off. So last time we spent probably, from your perspective, much too much time talking about conjugation. So thankfully we're done with that topic. And now we'll move on to transformation and then transduction. So transformation is, by definition, the uptake of free DNA from the environment. And so, as I've mentioned a couple times, probably most of you have at some point done a transformation in the laboratory, plasmids, into E. coli. Now, that is a very unnatural situation on multiple levels. One, plasmids don't typically transfer between cells by transformation. They usually go by the process that we talked about in the last lecture, conjugation. The other unusual thing about the type of transformation that you do with E. coli in the laboratory is that the DNA that's taken up from the environment in a transformation of E. coli is double-stranded DNA. So you introduce a plasmid, a double-stranded DNA plasmid, to these chemically treated E. coli cells, and it takes up double-stranded DNA. That's not what real transformation in the real world looks like. So transformation like that is biotechnologically very, convenient, widely used. But bacteria like E. coli actually are not naturally competent. They are not naturally capable of taking up foreign DNA. That's the definition of competence, the ability to take up foreign DNA from the environment. E. coli, on the other hand, can be made to do this. It can be tricked into do it in two ways. Classically, and still widely used, is to treat E. coli with something like calcium chloride that pokes holes in its membrane so that it can take up double-stranded DNA. Another way to do it is to put E. coli at high voltage, so you can shock it or do something called electroporation to get DNA into E. coli. But that's not part of E. coli's natural repertoire. But a large number of bacteria that exist in the environment and exist in association with us as human hosts do take up DNA naturally. And so, as I mentioned, humor is not very good in microbiology, but Mark is feeling rather incompetent today. I mean, this Mark, not that Mark. He's always competent. Yeah. So let's talk about how it is that this DNA is taken up. And so these are two different bacterial examples of well-studied competence systems that take up DNA. On the top is the gonococcus, Neisseria gonorrhea, the bacterium that you're going to spend the rest of your lives trying to avoid. This is the bacterium that causes gonorrhea. And actually now there are strains, sadly, in the community that are nearly untreatable by antibiotics. So very important to understand this bacterium. But DNA uptake in this particular case is going on by this protein complex shown on the top. You can tell this is a gram-negative bacterium because of the outer membrane and the periplasmic space. And you can see that this is a very complex structure designed to take DNA up from the environment. But one of the things that you notice about that is only one strand of the double-stranded DNA that's in the environment actually gets taken into the cell. It's the same thing if you talk about the gram-positive system shown in the bottom. The soil bacterium Bacillus subtilis, also very good at being naturally competent, capable of taking up DNA. In that case, what comes into the cell is also a single strand. This is in some, oh, I'm sorry, question?
Sorry, yeah. For the single strand that comes into the cell, how does it know, like, if it does have a medical or not to recognize it as foreign itself? Like, why doesn't it just, like, degrade the foreign DNA that's, like, content?
So how does the bacterium recognize this as self or foreign? So that's a really good question. So part of it is that methylation. So ideally, when these cells are taking up DNA from the environment, The idea is, in most cases, to take up DNA from other cells from the same species. And so being able to recognize a methyl group on a particular palindrome, a particular sequence, will mark that as self. But another reason for taking it up as single-stranded DNA as opposed to double-strand is also a self-defense mechanism. If this were viral DNA, taking up both strands would be definitely disadvantageous because now that DNA could be expressed. In this particular case, taking it up as single-stranded, if it happens to be something like a virus, it's less likely to be able to cause an infection in the cell. But the bacteria do have means to recognize self versus non-self DNA, and it does have to do with that methylation. But yeah, so you led into my next point perfectly. It brings it in as single-stranded DNA in part to protect itself. And oftentimes this DNA is being brought in not simply as a means to increase genetic diversity, but it can also be brought in to be broken down into nucleotides, so as a resource. So some bacteria will break down DNA in the environment or break down DNA intracellularly that it brings in as a source of energy, as a source of carbon, or as a source of nucleotides. But for the most part, when we're talking about competence at this level, in this class, we're talking about the uptake of genetic material for the express purpose of expressing that DNA in a new situation. So many bacteria have evolved ways, and we'll go over the details of this, to take up DNA at only certain times in their lifestyle or in their life cycle. So as I mentioned in reference to that question, many times bacteria don't want to take up foreign DNA from the environment because it may be deleterious. So a bacterium, like we'll talk about Streptococcus pneumoniae, has the ability to control competence by quorum sensing. So we'll go through the mechanism of this, but this bacterium will only take up foreign DNA when it knows that there are large numbers of other streptococcal cells in that environment. So if it then takes up DNA from the environment, there's a very good chance that it's taking up friendly DNA from kinfolk that have died and released their DNA into the environment. So these naturally competent bacteria actually use a couple of different strategies to make sure, as best they can, to take up DNA from their own cells. So one of those ways, as I just mentioned, is to control competence by quorum sensing. So Streptococci will use those autoinducer peptides as a census for when there are large numbers of streptococcal cells present. Then they'll take up DNA. There's another strategy that's been well documented in a gram-negative bacterium called Haemophilus influenzae. This was actually the first bacterial genome to be completely sequenced from one end to the other, or at least the first publicly released genome. And one of the things that people noticed when they started looking at this genome, there were these weird 11 base pair sequences that were scattered throughout the entire genome. Sometimes they were in coding sequences, sometimes they were in non-coding sequences. But they were perfect repeats and they were scattered throughout the chromosome. It turns out these end up being Haemophilus influenzae's way to specify its own DNA. So rather than do methylation to mark self from non-self, what Haemophilus influenzae does is that it recognizes by binding DNA in the environment to specific receptors and recognizing only DNA with that 11 base pair sequence, and then only taking up DNA that has that 11 base pair sequence, they can drastically reduce the frequency with which they take up foreign DNA. And by foreign here, I mean from a different species. Because this 11 base pair sequence, if you look through the database even today, you'll find that 11 base pair sequence in virtually no other species other than Haemophilus influenzae. So this bacterium has evolved to know, if you will, that when it sees that 11 base pair sequence, that's a sequence that's coming from that bacterium and not probably from a virus. So another really clever way to make sure that friendly DNA is being taken up. So this is the Streptococcus pneumoniae system that I'm talking about. So this is a nice system because it actually helps you review for the exam tonight that a handful of you will take tonight. The rest of you will take at some point this week. because this system ensures that Streptococcus pneumoniae, the bacterium that causes oftentimes ear infections in young children, but also pneumonia, and especially in elderly people. And some of you have this as your unknown in the laboratory this week. So as you're growing Streptococcus pneumoniae, think about this system whereby this bacterium has acquired the ability to take up DNA. So starting here at the top, you see that there is an export of a competence factor. So there's a protein synthesized in the cytoplasm of this bacterium called the competency factor, or CF. This peptide is going to be hydrolyzed, and a portion of the peptide is going to be exported. This is the autoinducer peptide. So thinking back to your studies on quorum sensing and gram-positive, this is the autoinducer peptide that we talked about, for instance, in Staphylococcus aureus. This actually ends up being a census, a surrogate for the concentration of cells in the environment. So as the concentration of this competence factor increases in density, it has the ability to now more frequently interact with this histidine kinase. So this is a two-component system, just like we talked about with the ONC and OMP-R system. But now the competency factor peptide binds to the histidine kinase COMD on the outside. Remember that this type of quorum sensing, the molecule does not re-enter the cell. This is different than homoserine lactone. Competency factor binds to COMD. It becomes phosphorylated on the cytoplasmic side as a histidine kinase. That phosphoryl group is transferred to an aspartate on the response regulator here known as COM-E. So COM-E is analogous to the OMP-R that we talked about in the E. coli system. Once that becomes phosphorylated, it now serves as a positive, an activator protein that will increase the transcription of one gene and one gene only. And it's a specific gene here called COMX, which encodes an alternative sigma factor. This sigma factor, again, so this is a beautiful review for the exam. If you can understand this, this is helping you study for the exam, even as you're sitting in lecture. This alternative sigma factor, now present only when the cells are at quorum, is now going to substitute in to the RNA polymerase to form a holoenzyme, which now and only now will have the ability to express the CLG operon, the cell operon, the CFL, and the COI operon. All of these operons are expressed only when SIGH, this alternative sigma factor, is present, which is only present at quorum, when there's lots of streptococcal cells in that environment. All of these genes are transcribed, all of these operons are transcribed and translated into proteins to make that very complex transposome, transformosome, shown here, this multi-protein complex that has the ability to take DNA from the environment, bring in a single strand, degrade the other strand in the process of doing it. So very highly evolved, very efficient way to take in DNA at only the appropriate time in the life cycle of that streptococcus. Okay, that's all I want to say about transformation. It's probably more than you ever wanted to know about transformation. But one of the things that I'd like you to remember, big picture, beyond this class, is that when you do a transformation in the laboratory and you're transforming E. coli, you're not actually doing a transformation the way transformation evolved to occur. You're just taking advantage of somebody's clever work showing that, hey, we can poke a hole in E. coli and poke in double-stranded DNA. It's a very artificial system. So now let's move on to the third form of bacterial DNA transfer between cells, transduction. So transduction, by definition, is the transfer of DNA by means of a virus. So simply transferring viral DNA to a bacterium is not transduction. That's an infection. What transduction really means is to spread bacterial DNA kind of accidentally, if you will, by means of a virus. So just spreading viral DNA is not advantageous or beneficial to the bacterium. But the ability to spread bacterial DNA by means of viruses has the ability to generate more genetic diversity in a bacterial population. And so it ends up being a good way that bacteria can evolve quite rapidly. So before we talk, and we're going to talk in nauseating detail about bacteriophage and viruses later on in the semester, but we need to, if we're going to talk about transduction at this point, we need some background on how bacteriophage work. So bacterial viruses, or bacteriophage, or as more people than often pronounce them, bacteriophage, which I think is incorrect, but I'll always lose that argument. Bacteriophage tend to have one of two different lifestyles. So if you take all of the bacteriophage entities in the world, they tend to fall into one of two flavors. They're either referred to as lytic, or actually I prefer the term virulent, or they're temperate, sometimes referred to as lysogenic. But I think temperate is the better example. We're going to talk a lot about lambda later on, and we're going to introduce lambda a little bit this morning. So here is a very generalized scheme shown at the top of what a bacteriophage or bacterial virus looks like. The red represents the double-stranded DNA genome of this particular virus. The phage binds to a specific receptor on the cell of this bacterium and then injects its DNA into the cytoplasm of that bacterium. Now, at this particular point, under just normal laboratory conditions, healthy conditions for the E. coli, what's going to happen is that viral genome will circularize, it will begin to replicate itself and to produce all kinds of proteins that make up new baby viruses, including a lysis protein that's going to burst the cell and release maybe 50 or 100 cute little baby viruses from the cell. So this is what's known as the lytic life cycle. There are certain viruses that can do nothing more than this lytic lifestyle, T4 and T7. Viruses like that simply have the ability to infect the cytoplasm of the cell, replicate their genomes, express their proteins, lyse the cells, and go on. Very efficient. But what happens if that lytic virus puts its DNA into a bacterium and then realizes, uh-oh, this bacterium's in trouble. This bacterium doesn't have very much energy. It doesn't have a lot of amino acids. It's really struggling. This is a sick bacterium. Well, in that particular case, the virus is really, it's kind of cast, the die are cast, because viruses separate their genetic material from the virion, and they can't suck it back out. So if this virus injects its DNA into a host that's really not doing very well, sorry, thanks for playing, but you're probably not going to generate new viruses because there's just not enough resources there. So this is a disadvantage to that type of a lifestyle, the purely virulent lifestyle. And so because of that, I think probably the more common forms of bacteriophage that are found in the world are what are known as temperate bacteriophage. These are viruses that have the ability to make a choice after they've infected a cell. So if this virus is a temperate virus, like lambda is a temperate virus, it injects its DNA into this cell. If the cell is fat and happy, fine. It will just immediately start to express the lambda genes after it circularizes its genome, lyse that cell, and release 100 little baby lambda. But if the cell is struggling, if the cell has some kind of nutrient deficit, what the virus can now do is it can make the decision to wait. It can wait things out. It can't suck that DNA back out into the virus, so it really has no choice but to incorporate its DNA into the genome of that bacterium and form what's known as a lysogen. So now the viral DNA incorporated into the larger circle here of the genome of the bacterium, that viral DNA is referred to as a prophage, and the bacterium itself is referred to as a lysogen. Now, if that bacterium happens to survive whatever hard times it's found itself on, great. The virus just replicates along with the chromosome. Every time this lysogen chromosome replicates, it's going to pass on a copy of the prophage to the daughter cell as well. And many bacteria in the environment are lysogens for numerous viruses. You can think of them really as ticking time bombs, because at some frequency, this cell may just divide and divide and divide, and everybody's happy. But then all of a sudden, if something just triggers that cell to perhaps indicate that it might be going to die. In this particular cartoon, they're using UV light as a damaging agent. The virus, the prophage, even though it's just DNA at this point, retains the ability to detect when that cell is going to die. It's a really clever, really evil kind of a pathogenic mechanism. When DNA damage occurs to this bacterium, or really antibiotic therapy can do this too, but anything that tends to damage that DNA is a signal to the virus that its host might die. And so if it's going to hope to survive, it has no choice but to then resurrect itself from the chromosome, circularize itself, and try its best to produce baby viruses and lyse that cell before the cell dies. Because if it doesn't do that, if that cell dies because of whatever stress that it's under, the virus is dead with it. So that DNA is lost, that prophage will be dead. And so thinking back to our discussion on F plasmid and the formation of high-frequency recombinant cells, we talked about the fact that the F plasmid was an episome. It had the ability to exist in the chromosome as a circular DNA or incorporate into the chromosome. So as you think about things and you're studying for exams, think about some of the similarities between what we talk about with the F plasmid and viruses. So, and remembering always my favorite hypothesis on plasmids is that they are really nothing more, I shouldn't say nothing more, they are really highly evolved viruses. This is another example where you can see the similarity between how conjugative plasmids like F plasmid can behave very much like a virus. They can go into and out of the chromosome. So this is called induction. So this is the virus coming out under the insult to that cell to resurrect itself to produce viruses. So this decision to utilize the lifestyle, whether it's lytic or lysogenic, depends on the environmental factors. Most definitely, or most especially important, is the nutritional status of the cell. Cells in poor nutritional condition tend to become lysogens. The virus tends to just insert its DNA and wait for better times. Now, how does that relate to the transfer of bacterial DNA to other bacteria? So now we want to talk about transduction, the transfer of DNA from one bacterium to another. And so we're going to talk about two different types of transduction, specialized transduction and generalized transduction. So specialized transduction carries only a small portion of the restricted section of the bacterial genome. It's limited to just those areas of the DNA on either side of the prophage. We'll talk about this specifically in the case of lambda in a minute, but just in general broad brushstrokes, what you see here is that here is a lysogenic bacterium. It has a prophage in its genome. Now it goes through induction, something that damages the DNA or threatens the life of the bacterium. It tries to resurrect itself from the chromosome and does. And more often than not, it resurrects itself as a perfect circular viral genome and tries to produce viruses and gets out of town. But at some frequency, especially because there's large numbers of cells involved here, stuff happens. And so what happens is a misexcision. So during the deintegration of that of that viral genome, you'll see that only part of the viral genome comes out, and it brings with it part of the bacterial chromosome. That part that it brings with it has to, by definition, be immediately adjacent on one end or the other, the right-hand side of that prophage or the left-hand side. This DNA shown here in blue on this smaller circle is the DNA that now can potentially be passed to a different bacterial cell. This is why it's called specialized transduction. A portion of the chromosome 180 degrees across the circle here has no chance at all of being transferred to another bacterium by specialized transduction. The DNA has to be adjacent to that prophage. So because of that misexcision, some portion of the genome now gets packaged into these viruses. So you'll see these chimeric genomes that are partly virus, partly bacterial. that lyse from that cell. Now, that virus is almost certainly defective, because in this particular cell where this occurs, notice that all of the viral genes are still present. It's only that some of the virus genes are on the chromosome and some of the virus genes are on that circular form. But in that particular cell where that misexcision occurred, every viral gene is present. So it can replicate its genome, it can produce all of the bacteriophage coat proteins for the capsid, it can lyse that cell and release these viruses into the environment. But notice that now what's packaged into these cells is only a part of the viral genome. A part is left behind because it was part of the bacterial chromosome. So these particles are almost certainly defective viruses. But if they are still infective, they still have all of the coat proteins necessary to infect another cell and to inject that DNA into that cell. But this cell will probably never undergo lysis from that virus because there's just not all of the genetic material of the virus that came over to this second cell. But what that second cell did get was some portion of DNA, shown here in blue, which has the possibility of crossing over. So homologous recombination mediated by REC-A. If there's similar sequence between that piece of DNA coming from the previous bacterial host and the new host that's just been infected, it can exchange that DNA. So it can add in new genetic sequences, which may add to the variability of that cell, which may increase the evolution or increase the fitness, rather, of that particular cell. But that cell is not actually going to produce any more viruses. It cannot do that. So this is a more specific example of how this occurs in lambda. So lambda is the paradigm for temperate bacteriophage. It's where most of what we know about, well, even for that matter, about retroviruses, because it has a lot of similarity to human retroviruses. But here is the circularized genome of lambda. We'll go over this in the virus section, but what's packaged into the head or the capsid of lambda is actually not circular, it's linear DNA. It becomes circularized upon infection, upon injection into the E. coli cytoplasm. But now, if this circularized genome is in a cell that's nutritionally challenged, and so the bacterium is going to go into the temperate or lysogenic phase of lifestyle rather than lytic, what it's going to do is it's going to use its attachment site, cleverly abbreviated ATT. The attachment site for lambda actually has a similar sequence and one position on the E. coli chromosome. There's only one place in E. coli where lambda can go in, and it's called the bacterial at site. It's shown here in purple. It's just downstream of the galactose utilization operon. And so this virus can insert itself into the chromosome, and so now what you see here is the formation of a lambda lysogen. This is a perfectly functional E. coli cell that can live forever, can divide. Every time it divides, it creates another copy of the lambda genome. So what you see now is that after time, what can happen is if this bacterial lysogen becomes stressed, if there's some damage to the DNA, the virus makes the decision to resurrect itself from the chromosome. So on the left here is what's shown would normally happen. From the virus's point of view, from lambda's point of view, this is exactly what it wants to happen, this normal excision. It will use those two attachment sites using a lambda gene called excisionase, appropriately enough, to recombine and to resurrect a perfect lambda circle, just like what came in generations ago, shown here at the top. All of the lambda genes are here in this circular piece of DNA. They've left behind nothing on the bacterial chromosome. And now this intact genome can produce all of the lambda proteins necessary to form or to replicate the genome, to form the capsid, and to lyse the cell. And so from lambda's point of view, this is a perfect situation. The bacterium maybe was going to die, so it resurrected itself, it produced new viruses, it got out of town before the cell died. Now, What can also happen is what's known as the formation of a lambda D-gal particle. D standing for defective, gal standing for the fact that it takes with it the galactose utilization operon. So it's not just trivial that this cartoon shows that this 3-gene operon that E. coli uses to utilize the sugar galactose, this is a catabolic pathway, it's adjacent to this. So what happens is there's a misexcision. So just for some weird reason during the excision process, lambda does a crossing over at a point where it shouldn't, something called illegitimate recombination. But what comes out is a circle. But now you'll see that there are more than 3 genes. Sadly, there are more than 3 genes on the lambda genome. But this cartoon, for simplification, shows 3 different loci. Think of them as 3 different genes on the lambda genome. And now you see that what's been resurrected has only genes 1 and 2. Gene 3 is left behind on the chromosome. But what has been brought up into this circular resurrected DNA is the galactose utilization operon. So this asymmetric or illegitimate excision removes an adjacent sequence. So again, we're talking about specialized transduction. So now you're seeing why it is that it's specialized. It can only be DNA that's on this side, on the left side, or on the right side of that genome. So, but in this particular cell where this event has occurred, all of the viral genes are present. So this cell that has just undergone this misexcision can still produce all of the lambda proteins necessary for lysis of that cell and packaging the lambda genome. But what's going to be packaged is going to be this genome, which only has lambda genes 1 and 2, but also has this galactose utilization operon. So this can now be passed on to another cell. So here is the result of infection of another E. coli cell with this lambda D-gal particle. So here, it's come out of the genome, it's left behind gene number 3. What goes into another cell is just lambda's genes 1 and 2, which by themselves are not enough to do the infection. But it did bring over a galactose utilization operon. In this particular example, they're showing an example where the cell that is now infected has a mutation in its native galactose utilization operon. So here it's got a defective gene somewhere in that 3-gene operon so that it can't use galactose. So you can return a functional galactose utilization operon to this cell because now by crossing over, it's bringing a functional galactose utilization operon to that cell. That's a very artificial laboratory situation where somebody deliberately takes a galactose mutant and infects it with this lambda D-gal particle. But in real life, viruses, not just lambda, are transferring adjacent DNA, not just to other bacterial cells in the same species, but probably even to different species. So this is a significant way in which genes are transferred between cells. If you look back at one of the first slides in this slide set, I showed and talked a little bit about how the E. coli 0157H7, that hemolytic uremic syndrome, the jack-in-the-box E. coli that kills so many children from kidney failure. The reason or the mechanism behind the evolution of that particular killer strain of E. coli was exactly this. So a virus that normally doesn't infect E. coli, that normally infects Shigella, brought with it the toxin gene for a toxin produced normally by Shigella and put that toxin gene as part of its genome into this new host. So it transferred DNA from a completely different genus into E. coli. So this is part of the reason why it's important that we understand viral transduction, because it oftentimes can have significant effects on the phenotype of cells that survive the infection. So this particular cell the stable transductant would have a functional galactose utilization operon, so its phenotype would be significantly changed. We're not going to go through this mess on the left. This is very much a laboratory artifact where you can artificially infect a lamp. This probably happens at some frequency in nature, but it's more of a laboratory thing. So it's not worth our time to go through that. Now, I seem to have somehow missed a type of generalization that I wanted to talk about. I should know my own slides by this point in my career, shouldn't I? We didn't talk about generalized transduction. How did I manage to lose some slides? Oh well, let's just move on. And hopefully in another lecture I'll find my slides on generalized transduction. Because generalized transduction, just because I know you're curious about it, has the capability of taking any piece of bacterial DNA from the infected cell and packaging it instead of the viral genome and spreading that to another cell. But hopefully we'll get to that in another lecture. But in the meantime, let's move on with whatever slide set I happen to have selected to use today. We're going to talk now about insertion sequences and transposons because they use a different kind of recombination called site-specific recombination. The recombination events that we've been talking to up until this point are driven by Rec-A and driven by DNA sequence homology between the two pieces of DNA that are being recombined together. In site-specific recombination, best exemplified by transposases utilized by insertion sequences and transposons, there's not necessarily DNA sequence between the two pieces of DNA that are being joined together. But there is specificity, so there is a target sequence that comes in. So we've already talked, I introduced the concept of insertion sequences already. So we talked about in the F plasmid, the F plasmid had several copies of insertion sequence 1, insertion sequence 2. These are genetic parasites, just plain and simple, but they are very simple parasites. Insertion sequences, shown here on the top, exist as nothing more than DNA sequences that have inverted repeats, the IR stands for inverted repeats, on either end, flanking one gene, the transposase gene. This transposase gene encodes an appropriately named enzyme, transposase, which has the capability of binding to these two inverted repeats and binding to a target sequence on some other sequence. In this particular case, they've hypothetically shown AATTC as the target site for transposition. Transposase will now insert a copy of that insertion sequence into there, and that's it. That's the whole lifestyle of an insertion sequence. Really a pretty boring genetic parasite. There's not a lot of phenotype associated with them. They don't carry antibiotic resistance markers with them or anything like that, so they don't have a lot of significance for those of you interested in biomedical concerns. The only things where you can occasionally see phenotypes is if by random chance an insertion sequence like this lands in some type of a gene and interrupts it. So if the loss of that gene leads to a phenotype, then you can see a phenotype associated with this movement of an insertion sequence. So insertion sequences are very simple. They're just the inverted repeats used in the transposition. Only have the transposase to bind to those inverted repeats. Transposons, on the other hand, are a lot more interesting. They're a lot more complicated, and they have a lot more real-world applications. So in both cases, so here is an example, a hypothetical example of what a transposon would look like, a composite transposon. So transposons oftentimes have, or not oftentimes, they have two insertion sequences, one on each end. Now, these insertion sequences can be independent actors. This insertion sequence with its transposase here and inverted repeats shown by those yellow boxes, even though they're not, well, they're labeled on the left-hand side, can use this transposase in between it to move or move this inverted repeat to some other region. But what happens a lot of times over the course of evolution is that mutations, random occurrences occur such that this inside inverted repeat becomes damaged. The DNA sequence is changed in that inverted repeat. And if it's changed enough that the transposase encoded here can't recognize that right-hand side inverted repeat, that entire insertion sequence is in essence dead. it can never move. The transposase has to be able to bind to both of those internal or inverted repeats in order to move it. But there's good news if you're the insertion sequence. If there's another copy of the insertion sequence somewhere nearby, there's a possibility that that same transposase, even though it can't bind to this right-hand internal inverted repeat, if it can bind to this external one, the one farthest to the left, as well as to the one furthest to the right, now the movement by that transposase is not just moving the transposon gene, it's moving everything in between it. So a lot of times what has happened is that these insertion sequences have lost some type of sequence identity in these inverted repeats, so that now the transposase is recognizing only the outermost inverted repeats. And so now what moves? Everything in between. This is why these transposons are so much more important, because they have the ability to mobilize genes in here. And you'll see that a lot of the times the genes that end up by natural selection being moved into there are antibiotic resistance genes. This is part of the reason why transposons were discovered in the first place. But in either case, whether we're talking about transposons or insertion sequences, the way that this mobile piece of DNA moves into another location is that the transposase has a target sequence, such as this AATTC, a target sequence. It's really neat because you can just do what's called sequence gazing. You can just look at DNA sequences. You can do it by eye, but it's easier to use some type of an algorithm. And it's easy to spot transposons or insertion sequences because when an insertion sequence goes into this sequence, it duplicates it. So here, shown below, is the end result of either an insertion sequence or a transposon going in. Here's the target sequence, and notice that it's duplicated on each side. These are direct repeats. This is different than an inverted repeat. I love this slide because it shows in proximity what a direct repeat looks like and an inverted repeat. And again, in terms of studying for the exam tonight, or the rest of this week, here's another excuse to look at inverted repeats. So here's the left-hand end of the inverted repeat of transposon 3. On this top strand, read 5' to 3', GGGGT. Look at the opposite strand, because that's the strand that's 5' to 3', GGGTC. You see the inverted repeat. It has to be on the other strand. Now, so that's from here, from the beginning of that inverted repeat to the end of the inverted repeat. That's the transposon, or that's the insertion sequence. The sequence just adjacent to it is the duplicated target, AATTC. But look at where the duplication is. This is a direct repeat because on the same strand, and still 5' to 3', AATTC, on the right-hand side, AATTC. You don't have to go to the other strand. This is a direct repeat. So, it's, in terms of, if for no other reason than to get questions correct on the exam, please make sure that you understand the difference between and how to recognize direct repeats versus inverted repeats. Here's my generalized transduction stuff. So recombination has occurred within my PowerPoint slide and has changed the order of my slides. I'm going to skip that for the moment. I promise in the next lecture, I'll get this straight. Let's continue on talking about mobile elements and these insertion sequences or transposons. So again, the insertion sequence is the simplest example of mobile DNA element. Just has the inverted repeats and the transposase gene. You can have a simple transposon where the ends of the transposon don't have individual insertion sequences, like in the last slide that I was just showing. Here, you have inverted repeats that flank antibiotic resistance genes, the transposase gene, and quite frequently a resolvase gene. We'll talk about the function of resolvase gene when we talk about replicative transposition and how this transposon can copy itself and increase its copy number. And then there's the composite transposons that I was just talking about, where you have different copies of the same IS element, and that's an important point. These two IS elements have to be, at one point, have been identical, because the transposase encoded by this element will recognize only its own inverted repeats. And so it's not going to recognize these other inverted repeats unless they're identical. And so again, oftentimes mutations have occurred at the internal inverted repeats, so that now the transposase rather, recognizes the outermost insertion sequences and now moves everything, which oftentimes includes these antibiotic resistance genes. So again, just to show you a reason why we want to understand these transposons, we've talked a little bit about, you already understand a little bit about vancomycin resistance and how difficult it is to accomplish. So you actually answered a question, many of you, on the last exam, on some of the different ways in which a vancomycin-resistant bacterium has to differ in order to be resistant. And part of the reason why vancomycin resistance was slow to evolve was there are so many different changes that have to occur to a bacterium that a bacterium would have to acquire a whole suite of different genes. Well, here they are. Every freaking one of them is contained within one transposon. So now, rather than have all of these individual genes scattered throughout the chromosome of one bacterium, making that one bacterium vancomycin resistant, over the course of time and under the selective pressure of us using too much vancomycin, random rearrangements have occurred so that all of these genes have now been picked up, have been vacuumed up into a single transposon. So here is transposon 1546. This was the first transposon isolated in 1988 from a bacterium, Enterococcus faecalis. Again, a bacterium that many of you have as your unknown in the laboratory this week. None of you have one that's resistant to vancomycin, I promise you that. But that particular bacterium has a transposon, which is now spread to other species of bacteria. This is why we don't want to use vancomycin unless we have to, because the possession of this transposon is only advantageous if the bacterium encounters vancomycin. Otherwise, it's kind of excess baggage. So this is why we don't want to use vancomycin. But all of these genes, any one by themselves is completely useless in terms of vancomycin resistance. And then, of course, the other genes that it has to have are the transposase and the resolvase to help during the infection of other cells. So really, when we talk about antibiotic resistance in more detail later in the class, we're going to come back to this and talk about how each of these molecules works in vancomycin resistance, because it's really, really cool. Two of these genes, VAN-R and VAN-S, you know something about already. This is a two-component signal transduction system that senses, guess what? Vancomycin. This is a sensory protein that has as its environmental ligand the antibiotic vancomycin. When it senses vancomycin, it phosphorylates this response regulator, and guess what that response regulator does? It transcribes these other genes to now begin to express the bacterium's ability to resist vancomycin, only when vancomycin is present. This is you couldn't design something like this if you were a science fiction writer, any better than the way nature has allowed this to evolve. Okay, and then the last point that I want to make in this lecture, this is more just to make myself feel better, because we spend so much time talking about so many different genes. This is a paper that came out about 6 years ago now, where somebody looked at all of the genomes and metagenomes that had been sequenced up until that date. So many more have been done now. And if you look at the number of genomes done and the abundance of particular classes of genes, it's almost a one-to-one. It's not a very good R, but look at some of the genes that fall way off of this strategy. Transposase is probably the most abundant gene in bacterial nature. But also other things that we've looked at, two-component signal transduction proteins, are way off the chart. ABC transporters, ATP binding proteins, transcriptional regulators, methyl-accepting chemotaxis proteins. So the proteins that we've spent most of the time talking about end up being, because I designed it that way, being the most abundant bacterial proteins in the microbiota in the world. Okay, on that self-congratulatory note, I'll see you guys on Friday. Don't forget the exam tonight or some point during the week.
You